Hello friends, today we are going to discuss how to identify the cell in DLC practical. So we will be identifying the cells and we will be also able to draw the diagrams of various leukocytes. So here if we see, first point is size by which you can differentiate about various cells. So whenever you are drawing, so you have to draw a proportionate diagram. So three circles I have drawn. One big circle is there. One again small circle, little bit less than that of three circles, and one cell is this. So this is you can say a big cell which you can call it monocyte. This is the RBC with which you are going to compare all other cells in peripheral smear, and this is lymphocyte. You can call it as small lymphocyte. So what is the next point by which you can differentiate? So next point is our nucleus. Now just see, initially I have drawn here in this cell 1, 2, 3. 3-lobe nucleus, or you can say multi-lobe nucleus. Then here I have just drawn only 2 lobes of nucleus. Here again I have drawn only 2 lobes of nucleus. Now if you see here, there is a big nucleus is there. There are no lobes. And here again this nucleus size is again big, again there are no lobe. So if you compare nucleus to cytoplasmic ratio here, here and here compared to these two cells, so monocyte you can say it is almost equal or little bit less than 1, and this cell it is covering almost whole of the cytoplasm. Nucleus is covering whole of the cytoplasm. So till now we have discussed about size, nucleus, multi-lobe nucleus, bilobe nucleus, no lobe nucleus, also again no lobe of nucleus, and cytoplasm proportion is very high in this above three cells. Cytoplasm portion is you can say almost similar to nucleus, and here cytoplasm portion is very less compared to that of nucleus. So these are 5 cells. Now next we will be discussing what are the various granules. So again this diagram we have discussed. Now we will go to granules, and in the end I will be telling you which cells are nominated as neutrophil, basophil, lymphocyte, all that we will be discussing. Now just see here, I have drawn here, if you see first cell, just concentrate on this. I have drawn a very fine color granules. In your book, some book it is given as pinkish, but while drawing the diagram, many times we are drawing fine color granules of both color. So that is why I have drawn hemotoxin color as well as eosin color granules, but see carefully, all granules are fine. If you see next cell, all these granules are very big, or to better term is coarse. And what is the color of that? They are, you can say, reddish color, or to be precise they are eosinophilic color. So these was the other cells. Now just see here, again here granules are very granules you can see, but they are again coarse. So these are the 3 cells. Now here in this there are this cell as well as this cell, there are no granules. Now last portion is cytoplasm. In the cytoplasm, if you see here, because of eosinophilic granules, this cell will look as if pinkish or eosinophilic. So while drawing cytoplasm, you have to draw cytoplasm like this. Other you can draw faint color bluish granules in all other cells. So this is the way by which you can remember various other cells. So this was neutrophil, multilobe nucleus, fine granules, and size is almost 10 to 14 micron, or you can say 1.5 to 2 times that of RBC. Then eosinophil, coarse granule, coarse eosinophilic granule, bilobe nucleus, size again same that of neutrophil, that is 1.5 to 2 times that of RBC. Then basophil, see basophil, as the name suggests, it is basophilic. Granules are coarse and granules are so high that they cover the boundary of nucleus. You cannot see nucleus. So this is a basophil. Then monocyte, which is having a highest size, almost you can say around 2 to 2.5 times that of RBC. And this is lymphocyte. Lymphocyte, depending on size, you can divide it as small lymphocyte or large lymphocyte. If it is little more than that of RBC, then it will be small lymphocyte. If it is say 1.5 times that of RBC, then it will be large lymphocyte. So these are the simple way as well as the way by which you can draw various diagram and as well as identify the cells. So to summarize, monocyte is the largest leukocyte. Fine granules are seen in neutrophil. Coarse granules are seen in other basophil, eosinophil, but color is different. And multilobe nucleus is observed in neutrophil. So for all this practical, you have to practice, you have to use microscope. More you practice, more you will be able to identify the cell properly. So thanks